Engaging in regular napping has been found to be associated with a greater brain volume. Greetings and salutations to everyone. In today's video, we will be exploring the benefits of daytime napping in preserving brain health as we age. A recent study conducted by researchers at UCL and the University of the Republic in Uruguay has revealed that regular napping can slow down the rate at which our brains shrink, ultimately leading to larger total brain volume. This increase in brain volume is associated with better brain health and a reduced risk of dementia and other diseases. To establish a causal relationship between daytime napping and brain health, the researchers utilized a technique known as Mendelian randomization. It is worth mentioning that this study received support from Diabetes UK, the British Heart Foundation, and the Diabetes Research and Wellness Foundation. Without any further delay, let us dive into our discussion. A recently published study in the journal Sleep Health examined data from individuals between the ages of 40 and 69, revealing a direct correlation between regular napping and increased total brain volume. This increase in brain volume is an indicator of optimal brain health, which is associated with a decreased likelihood of developing dementia and other illnesses. According to Dr. Victoria Garfield, the senior author from the MRC unit for lifelong health and aging at UCL, their research indicates that short daytime naps could potentially play a role in maintaining brain health as individuals age. Prior studies have indicated that taking a nap can bring about cognitive advantages, as individuals who engaged in a brief period of rest demonstrated superior performance in cognitive assessments during the subsequent hours compared to those who did not nap. The primary objective of the new research was to determine whether a cause and effect connection exists between daytime napping and the overall well-being of the brain. By employing the method of Mendelian randomization, researchers examined 97 segments of DNA that are believed to influence individuals' propensity for regular napping. They conducted a comparison between individuals who possess genetic variations associated with a higher inclination to nap and those who lack these variants. The study utilized data from the UK Biobank study, encompassing a sample size of 378,932 individuals, and assessed measures of brain health and cognitive function. The findings revealed that, on the whole, individuals genetically predisposed to napping exhibited a greater total brain volume. According to the research team's estimation, individuals who regularly nap exhibited a notable difference in brain volume compared to those who did not, equivalent to an aging effect of approximately 2.6 to 6.5 years. However, the scientists observed no discernible disparity in the performance of habitual nappers on 3 additional assessments of brain health and cognitive function, specifically hippocampal volume, reaction time, and visual processing. In this groundbreaking study, Valentina Paz, a PhD candidate from the University of the Republic in Uruguay and the MRC unit for lifelong health and aging at UCL, takes on the challenge of unraveling the intricate connection between regular daytime napping and its impact on cognitive function and brain structure. Unlike previous research, this study utilizes a unique approach called Mendelian randomization, which allows for the examination of genetic factors present at birth and eliminates the potential influence of confounding variables that may arise throughout one's lifetime. The findings of this study provide compelling evidence of a direct and causal relationship between habitual napping and an increase in total brain volume. Dr. Garfield expressed his hope that studies like this, which demonstrate the positive impact of short naps on health, can contribute to diminishing any remaining social stigma associated with daytime napping. In a previous study analysing information from 452,633 individuals in the UK Biobank, researchers successfully pinpointed the genetic variations that impact our tendency to take naps. The study, spearheaded by Dr. Hassan Dashti from Harvard University and Massachusetts General Hospital, who is also a contributor to the current study, identified these variants by relying on self-reported napping data, which was further corroborated by objective measurements of physical activity captured by a wrist-worn accelerometer. The researchers conducted a thorough analysis of health and cognition outcomes in individuals with these genetic variants, taking into account various subsets of these variants to ensure unbiased results. They specifically avoided including variants associated with excessive daytime sleepiness to eliminate potential bias. A total of 35,080 individuals from the larger UK Biobank sample had both their genetic data and brain scans obtained through magnetic resonance imaging, MRI, available for analysis. The authors acknowledged a limitation in their study, specifically that all participants were of white European ancestry, which may restrict the generalizability of the findings to other ethnicities. Although the researchers lacked data regarding the duration of naps, previous studies indicate that shorter naps, specifically those lasting 30 minutes or less, offer the most favorable cognitive advantages in the short term. Additionally, napping earlier in the day poses a lower risk of interfering with nocturnal sleep. A study conducted in both the UK and the Netherlands discovered that approximately 1/3 of individuals aged 65 and above had established a consistent habit of taking regular naps. Financial support for the study was provided by various organisations. In the United Kingdom, Diabetes UK, the British Heart Foundation, and the Diabetes Research and Wellness Foundation offered their support. In Uruguay, the Programa de Desarrollo de las Ciencias Básicas, PEDESIBA, MEC-UDELA, Agencia Nacional de Investigación e Innovación, ANI, Comisión Sectorial de Investigación Científica, CSIC, UDELA, and Comisión Académica de Posgrados, CAP, UDELA, provided funding. Additionally, the National Heart, Lung and Blood Institute in the United States contributed to the study. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.